presentation. I hope everyone can hear me okay. Let me know if there's any problems. And I'm going to start to share my screen here, and let's see if that works well. Let me see here, and so let's see if I can get this to work. Right. Okay, great. Well, again, it's a real pleasure for me to have the opportunity to do this presentation again this year. I'm sorry, like many of us, that we can't do this in person and have a chance to talk. But I think that it does also give the advantage that a lot of people who might not be able to come to the meeting in person will be able to, be able to participate in all these events. So I certainly think that the Foundation's done a great job with that. I just wanted to start, before I get into my main talk, to tell you a little bit about how the COVID epidemic or pandemic is affecting us all in research. And I think that probably it's similar to what several of the other speakers have dealt with. And it's been difficult this last year because many of us had to close our research labs, had to close our clinical trials, and weren't able to see patients or do research for at least 3 to 4 months. Fortunately, we're gradually ramping back up, but even with the improvements that we've been able to do in terms of safety working at the NIH, we still haven't been able to see any patients in person for about 6 months now. Hopefully that will change soon, and those of you that are enrolled in our studies and that are just coming for consultation will be able to do so shortly. The NIH is going very slowly and cautiously because they don't want to have any outbreaks that could really have an adverse effect on a lot of our patients, many of whom throughout the NIH are immunosuppressed and are severely affected with diseases like cancer or immune problems. So we really do have to be very cautious as we open back up our facilities. But I think the good news is that our lab, and I hope the labs of the speakers before me and others working on HSP, it's improved, and we're able to get back gradually to the efforts that are so important in making progress for this rare group of disorders. My talk is going to be a little bit more general than the other two speakers, but I thought they did a spectacular job of really presenting some of the points that I'm going to highlight, and they did so in a very detailed way, so I think it'll make it flow a little bit better, because I know you've already had a great introduction to a lot of what I'm going to be saying. Now, the spastic paraplegias, I know you've heard several good introductions to, but I just wanted to highlight a few aspects of it. I think both Peter and Darius mentioned it just now. Of course, HSPs are defined by the symptoms that we see, and that's lower limb spasticity, which usually has those spasms that people feel, as well as a component of weakness. And it's typically progressive, though perhaps not in all different forms of HSP. There might be some that are more developmental, and some that maybe have a combination of developmental and degenerative mechanisms. It's due to, and that's sort of my next point, it can be due to either one of those. It can be that the axons develop abnormally, or that there's degeneration. Of course, that's very important to make that distinction ultimately through research in terms of how we treat these disorders. And of course, the length dependency is why the legs are affected. You see, the legs are sort of, it ascends. The legs are affected first. Rarely do patients have problems in their arms or anything above the waist, but certainly it can happen. But the legs and the walking disability is the main thing that I think brings most patients to ultimately to the diagnosis and to physicians. And again, we've already heard a nice discussion of how you can have pure or complicated forms. And of course, that's sort of in the pre-genetic era, where a lot of the clinical aspects were the main ways that this disease was defined. And of course, it's important to still understand those, because those can be symptoms that are very disruptive to patients. And certainly if we could improve those, can substantially improve quality of life. I think we might have heard a little bit about prevalence. We know that these diseases are rare. We know that some are common, as Peter mentioned. Some are very rare, as Darius mentioned. And then there's a lot in between those two. One of the really remarkable things about this disorder, when you think about it in the context of other inherited neurological disorders or other common degenerative disorders, is that we have so many different genes. And that really allows us to study the basic science of these proteins that these genes produce, and to really start to tease out the different mechanisms that are involved in causing HSP. And I think that if we can target those fundamental mechanisms, and I think as both Peter and Darius said, then we really have a chance of making a meaningful impact on the progression of the disease, not just on the symptoms that people see, but really stop the disease in its tracks. And that's certainly our goal. I think there's also several points were brought up about the genetics of this disorder. And again, I think that we recognize that not everybody has a diagnosis, and that's an important gap that we still need to fill. Now, I thought I'd just go through my talk in 5 main sections. And the first is going to be genetics. And I sort of foreshadowed that by showing that we have all these genes, and that what those genes can do for us is they can give us tremendous insights into what might be causing this disease. But we also hope, and I think it's gradually being shown, that there's not 90 different forms or 90 different mechanisms. Because it would be very hard to imagine that we could really get enough momentum in any one of them if there were so many different causes. So our hope has always been that by having so many genes, we'd be able to learn and understand common themes that might be targets that might benefit lots of different types of HSPs. And of course, one of the greatest advantages that we've had in terms of finding those genes and sort of developing those common themes has been the real explosion in our understanding of genetic disease. And I like to show this figure as an early slide in most of my talks, because it really emphasizes what happened sort of in the middle of the last decade, and just after the turn of the century, around 2001, it was very expensive to sequence DNA. Now, however, it's down like many, many orders of magnitude in cost, from $8,000 or so in 2001 to something like 8 cents in 2014 to sequence 1 million base pairs of DNA. And of course, that lower cost has tremendously improve the diagnostic yield. We can test a lot more genes, and we can figure out, many more patients and diagnose many more patients. However, it comes with a bit of a caveat, I think, that Peter mentioned as well, is that most mutations don't cause disease. We all have thousands and tens of thousands of variations that make us unique. So one of the challenges, of course, is finding which mutations cause disease, maybe which ones just make us different. But what you'll notice is around 2007, 2008, when this cost came down dramatically, that's what's called next generation sequencing. It was a new way to sequence DNA, and it gave us the ability to, again, sequence DNA, but it didn't necessarily give us the diagnoses. And that's been through a lot of careful work by basic scientists and clinicians working together to understand which mutations are causing disease and which ones just make us different. And because of that, since I think I started studying HSPs around 2001, we were at SPG 15 or so, I think as Peter mentioned, now we're well above 80 different forms. So we've had this massive number of genes identified, including probably the most common forms that are already known. And so we can make a lot of progress based on what we know already. However, it's important to remember, I think the numbers vary from center to center, but probably at least 30%, 40%, some places even higher, of patients who we really think have HSP, we don't have a genetic diagnosis for. And it's important to understand the limitations of current testing. A lot of times it emphasizes what are called exomes, which are just the exons that make proteins. But it's important to remember that's only 1% to 2% of the genome. There's another 98% of the genome that often isn't studied in those tests or in those panels. So I think as time goes on and as sequencing gets even better and better, we will start to gradually fill in that gap. I think the encouraging thing is we have so many genes now, we can make a lot of progress toward understanding the pathophysiology and making progress toward starting trials. Even without more information. But I do think that for those of you who do have an HSP, maybe without a diagnosis, that over time that will improve. I don't know if it'll be quite as quick as that dramatic increase in the numbers we had, over the last 10 or so years, but I think that it will happen as the sequencing technologies get better. So I think that's one important aspect. We will still learn from new types of HSPs, new types of mutations as we get more genetic forms, but we really do have a tremendous advantage in the ones we already have. And that certainly allows us to understand the functions of HSP genes in different cells. And I've made diagrams like this many times over the years, even back about 10 years or so. And for the most part, the categories have stayed the same. We've been able to just add more genes to them, which I think is good because it means maybe we're on the right track in terms of how we categorize those. But it is important to remember that, proteins can be many different places. They can have many different functions. And as I think as Peter mentioned, we really have to keep understanding as much as we can about them, because not everything a protein does might be related to the disease. You know, it could be certain aspects of it. And that's, I think, where the sort of the convergence can help us, because if a lot of genes do the same thing and act together, or like Darius showed, they all bind to each other in a complex, it's pretty compelling that aspect of it is going to be very important, that functional aspect. And one of the things I'm going to do today is I'm going to really talk about two categories here. So again, as I think Darius and Peter talked about specific forms, I'm going to go a little more general and talk about categories. And the ones I'm going to focus on are in the lower right of my screen. And if I'm sharing my screen, you can probably see my arrow. One is tubular endoplasmic reticulum, and lysosomes, endosomes, autophagosomes. And again, I think that was brought up by Darius a bit, and Peter as well. You know, they're talking about organelles within the cell. And it's important to remember that neurons are cells. They're like other cells in our body, but they have some unique features. They communicate in different ways. In many cases, they're longer. Remember, a corticospinal axon that's involved in HSP can be up to 1 meter in length. And that's hundreds of thousands of times larger than most of our cells. So there's also this massive size that we have to consider as maybe why these cells are affected the most. One of the things that really jumps out when you look at the different proteins is that very, very few of them are only in the nervous system. And I think Peter brought that up as well. So why do we only have the nervous system involved? Is it just the cells are so big? Is that there's other factors that are combining with these mutations? And I think that's one of the things we really want to understand. And again, that's the reason we've heard so much about different model systems and so on. We need to study this in multiple ways to really understand what the mechanisms are and how that specifically relates to disease. But the advantage of having proteins that are expressed throughout the body, or at least broadly expressed outside the nervous system, is it gives us more chances and more different systems to study them. And that can make it a lot easier to understand how they function. And once we understand that function, it can help us move toward better ways to potentially treat these disorders. So I'm going to show a couple examples from our own work, but I'm going to try to mention, of course, what other people might be doing in the field as well, maybe in a more general way. So here's a group of patients we saw many years ago. Many of you may have SPG15 or SPG11, as I think Peter showed. SPG11 is the most common form of HSP that is recessive. So again, I think as we've heard already, there's different types of inheritance. You sometimes can have it where you have a parent and then a child, and it goes down from generation to generation. However, in this case, as you can see from A, when it's colored in dark here, it means you're affected. But the parents didn't have any symptoms, and 2 of the children did. And that's a case where it's what's called autosomal recessive. You need 2 copies of the gene that are mutated or changed in a way that creates dysfunction. And again, Peter gave a very nice description of loss of function versus gain of function. These types of recessive disorders are typically loss of function, because you have to have both genes gone. And you figure if one's mutated and it's acting in a dominant negative way or acting as a gain of function, you'd expect to have the parents having some symptoms. It's not 100% true, but certainly in this case, it makes sense that we've lost this protein. So much like the cases that Darius talked about, you know, when you think about therapy, you think about, well, okay, we want to understand this protein, how it functions, but we also think, is there a way we could possibly replace it? So that's, again, gets into different, as we understand the genetics, understand the functions, it also starts to get us thinking about how we might be able to treat this. Again, as Darius showed for some of his patients, you know, these patients have a thin corpus callosum, some white matter changes that are very typical, some changes in their eyes that you can see in the retina. So there's a certain group of symptoms that you often see with the HSPs. But it's important to remember that sometimes you can have one form and somebody with the same SPG, whether it's 11, 15, or any of the numbers, can have very different symptoms. And that's something we don't always understand, how there can be so much variability, sometimes even with the same mutation within the same family. And that's the kind of thing I think where the clinical studies, much like Darius showed, are really important. It's important for us to understand not just like what a few people that we might be seeing in our own practices have, but really if you take a lot of patients from across the world, you know, what are the common features? What are things that might be only present in a small subset of patients? And that can be very, very important for how you ultimately design a clinical trial. So again, it's important to understand the clinical aspects as well as how these proteins might be functioning and putting those two together. So here's an example, I think, that highlights what Peter said about that proteins are often expressed throughout the body, and yet we only say that it only affects the nervous system. But I think if you think about how it's affecting different proteins or different parts of the body, that's not always true. When we say symptoms, we mean that's what somebody complains of. They have spasticity, we see something abnormal on examination. However, if we take cells from patients with SPG 11 or 15, or probably other HSPs as well, and you look at them under a microscope, and you look at certain structures, you can see the abnormality. So even though we call it only affecting the brain, we see abnormalities in skin. It's just not to the point where it is clinically meaningful. It doesn't bother people. But it's important as we think about these disorders to think that these cells are affected, and that can be very helpful in terms of following responses to therapy or developing new models. Like in this example, if you look at panel A, 2-1 across the top is a normal sibling. And then the 2 to the right, 2 and 3, have SPG 15. And you'll notice that across the top, the endosomes look pretty normal. However, LAMP1, which is a marker for lysosomes, are dramatically different. They're much larger. So we can see an abnormality in somebody that only has a nervous system problem, but we can see the abnormality in a skin cell that when you look at it, it looks normal until you start looking at the organelles within them. And I think that gets back to Peter's point about how important it is for us to really understand the fundamental biology, because that's really in a lot of ways where the key to intervening in these diseases will lie. And again, I just give a summary of what we've learned about this, that there's a problem with forming lysosomes, or you develop these large autolysosome-like structures, and we can even see that in skin, and we're sure the same thing happens in the brain. Again, it's much easier in terms of studies to work outside the nervous system. Regular cells are easier to study in many ways, but we can learn a lot about what's probably the same process is probably happening in the nervous system that is happening in the periphery. So again, we can sort of put these pathways together. In the case of SPG 11 and 15, much like what Darius talked about with the AP complex, these proteins bind to each other. So again, it's already sort of evidence that there's this shared mechanism. In this case, it's almost definite because they can't function without each other. If you knock one down, the other one degrades. And that's probably why they are clinically almost identical. So again, I think in many cases, as we understand the biology, the clinical presentations that people have make a lot more sense usually. So again, I think that's one example of lysosome, endosomes, autophagosomes. Again, Darius talked about another one, a less common one, the AP4. But I think it's important to remember that many of those, though not all, are loss of function. So as we think about that, we can certainly screen for compounds that might suppress these abnormal phenotypes in culture systems or in model systems. But it's also important to remember as we think about therapy that, is there a chance that we could reintroduce these proteins some way through gene therapy or so on probably at a very early stage of life, so that we could, potentially prevent the problems from occurring. We don't know, but it's an important thing to remember with loss of function phenotypes, that's a different type of option that you might not have with a gain of function etiology. Now, the second one I'm going to talk about today is about endoplasmic reticulum. So again, that was just an outline of this sort of autophagosome, lysosome, endosome function. Now we have another one, which is endoplasmic reticulum. And in many ways, this is a particularly compelling one because so many proteins interact and do the same thing. And that includes the 3 most common forms. Peter mentioned SPG4, and there's, as he already introduced, M1 spastin and M87 spastin are 2 forms. So M1 is the form that's in the ER, M87 would be in other parts of the cell as well. And it also includes a protein called REAP1 and elastin1. And those are collectively 3 of the most common forms of HSP. But they're not just those 3. As we go on, more and more forms, and I think we have 2 more that I haven't put on the slide yet, are all related to mutations in proteins. Most of them are, like Peter said, are gain of function. So we have a mutant protein, though some are loss of function as well. And what these proteins all do is they actually form an organelle called the endoplasmic reticulum that is distributed throughout the cell. In fact, it's distributed throughout the cell so much that there isn't any part of the cell that doesn't have it. And one of my colleagues did a study where they studied where the ER was within the cytoplasm. And over 15 minutes, there was an ER tubule in contact with 97% of the cell. So the ER really is a sensor and effector. It does things and it senses things within the cell, to an extent that no other organelle can do. And it's also continuous. So you have a 1 meter neuron. You think about how small an organelle is, but it's 1 meter long in these cells. So again, you can imagine that anything that gets disrupted could have a rather severe effect on long cells in particular. Now, if you look at the ER, it has different morphologies, and that'll be more obvious in the next slide. We have this connected structure that has all these different shapes and sizes. It has tubes, it has sheets. But it's the largest continuous organelle in the cell. It's about 30% of the volume of your cell, maybe even a little bit more in most cells. And it's in all these different forms, but it's all connected into one large organelle. So if you're in the middle of one part, and if you're going down one of those tubes, you could eventually pop out in one of those sheets or some other part of the cell just by going through those tunnels. And that becomes important because if we think about why an organelle that's present in every cell in the body would be so specifically affecting these long corticospinal neurons, remember, part of this disease is based on length. It's the vast size of these cells that are, again, in many cases, hundreds of thousands of times larger than other cells in your body. So it's a massive difference in size. Now, we can understand a lot about how these different proteins work. In fact, many of them only go halfway through the membrane. So when you have a membrane, you have two leaflets, and proteins usually go through it. But in these cases, these proteins only go halfway through. So if you do that, what do you get? If you put a lot of those together, you get a wedge, and it makes a tube. It actually shapes the tube. So the tubular ER is caused in many cases by proteins that are mutated in the HSPs. And they're fused together into this network that we all see, even from high school biology. You see this vast network of ER. Many of those proteins are mutated in this disease, and it tells us how important that is. And you'll notice in panel B, as Peter was outlining so nicely, motors are very important, as are the microtubules, because the microtubules specify in many ways how the ER gets to where it's going. So if we think, well, ER is such a critical organelle to have throughout the cell, well, how does it get there? Well, it gets there with microtubules. So again, it starts to show how these themes might fit together. Peter was talking a lot about microtubules. I'm talking about ER, but remember, the two work hand in hand in many cellular functions. And in fact, the microtubules are really remarkable in their heterogeneity and what they can do. And we've recently finished a study, though these are not HSP proteins, where it shows that different parts of the ER get distributed in different parts of the cell by binding not just microtubules, but by binding specific types of microtubules. And these are differences in what are called glutamylation. It's a modification of microtubules that's very prevalent in the brain. But if you have one glutamate, it binds to one protein, and if you have multiple ones, it binds to another. And if you don't have any, a different protein binds. So again, I think it just emphasizes the close contact between the cytoskeleton, as Peter so nicely described, and the organelles within a cell. And again, it's very important that things are properly distributed. So we always talk about transport. How do we get something from somewhere else to somewhere else? But it's also important where they are at a given time. And in many cases, it's not just motors that move these things along. It's different proteins are interacting in very selective ways, particularly with microtubules in the nervous system, to keep things in the proper position. So again, I think that really emphasizes, I think, the point that was brought up earlier. It's really important for us to understand these functions at a very basic level, the very basic biology, again, as Peter mentioned multiple times, and that the more we understand that, I think the more we're going to see how things converge. We don't want to try to do it too soon. We don't want to move on when we really don't have that understanding, because again, as Peter said, we'll fail. If we pursue the wrong areas or wrong directions, the chance of success is low. Our chance of success is dramatically increased when these insights come together. And of course, an important part of that in terms of understanding the meaning of different cellular changes and the relationship to disease are models. And again, we've heard about several of those already. And again, these can range from flies to mice to all sorts of different types of models. In fact, they can even be plants. And when we first were studying some of these ER proteins years ago, it turned out that people had seen exactly the same thing we saw in neurons, essentially in a plant. This is actually a plant, and those are what are called root hairs. It's a root. And you'll notice on the right that the mutant form has short, impaired ones. So these are actually these long processes are individual processes off the cell, much like you'd see of the axons that Peter showed. And they're impaired in this mutant. And yeah, this was done long before we even started working on HSPs. So sometimes it's important to look back in the literature and understand that the fundamental biology is often shared among lots of different systems, and that it can be particularly valuable if you start seeing the same type of abnormality across different models, because then I think it makes it more likely that you're going to be correct. Something that's that highly conserved is going to be potentially important. And in fact, these plants don't grow very well. Many, maybe like the axons don't grow very well, and the long neurons don't function very well in HSPs. And in fact, we see the same thing. This was, as we've heard about, we can do these iPS cells, and we did this many years ago. You can take a cell from a patient, or now you can just gene edit a mutation in, to mimic what a patient might have. And you can make them into neurons. You can make them into different types of neurons by adding different proteins and different culture conditions. And you'll see that what you notice is that the patient on the bottom left here has short neurites, processes coming off of the cell bodies. And a wild type, a normal cell that was differentiated, has long processes. And again, by adding different drugs, and again, I think we heard you can screen for these, you can do lots of ways, but certainly we knew the importance of microtubules. So by adding different drugs that can affect microtubules, you can notice that in many cases you can get this closer back to normal. So I think it emphasizes the importance of model systems as a way to potentially test new ideas in terms of treatment. But of course, ultimately, we don't want to stay with just a cell-based system. We want to move gradually up to, model systems that are closer and closer to us. Now, so we can do things like flies. We didn't hear too much about those, but flies are very amenable to genetic manipulation, and they're very fast. You can get the data very quickly. And one of the things we noticed in a collaborative study with several different groups was that the same elastin protein that is involved in one of the common forms of spastic paraplegia caused abnormalities in lipid droplets, which are another organelle that comes off the endoplasmic reticulum. And in a fat body, you notice these big, huge fat lipid droplets here. In the mutant form, they're much smaller and disrupted. And if you look at triglycerides and things, again, that we hear about all the time in terms of human metabolism, we see huge differences. And actually the flies die very young. That's in contrast to people, because in many cases, when we look at simpler systems like Drosophila, they might have one protein that does something. One advantage we have as humans is we have many proteins that do the same thing. We have 3 elastin proteins. So in some cases, it can sort of do some of the functions if one of them is impaired in some way. So again, whereas it's more deleterious or more harmful to flies, in humans it has a relatively small phenotype. It's specifically the corticospinal neurons, where all the other neurons seem to be much more normal. But these do give us clues. So now we not only know the ER is involved, we not only know that long processes are involved, we know it has something to do with lipids. And certainly we can make other model systems and see if we get the same thing. So we know REAP is another protein that, like elastin, is involved in ER. And if you make a model system, much like Peter showed, you can get a normal mouse that walks along a treadmill. And if you have a mutant, this guy can't keep up because he can't move his rear legs. And we can actually record it even at 1 day old, before the mouse even, well, they don't really walk that much at that age, but certainly before they've really shown a lot of symptoms. If we put a little electrode into their spinal cord, we can actually see abnormalities, even at very early stages, electrophysiologically. And what we see is excessive activity. And that's what spasticity is. You feel this excessive activity and the spasms and the increased muscle tone. And that's due to, again, things like shorter latencies. I won't get into all the physiology, but essentially, I think the way to think about this is that models can tell you different things. and that sometimes a mouse, where the circuitry might be a little bit more similar to a human, can tell us a little bit more about the physiology, where something like a fly or a plant or even yeast cells can tell us maybe a little bit more about the fundamental biology. So again, it's important to remember that each model system has its strengths, and we can learn a lot from different types of systems. Oops. And in fact, if we look at these mice and we open up their chest cavity, we see fat tissue in a normal mouse, and we see it's almost completely gone in these mutant mouse at a very early age, even at just 2 months before they have much in the way of clinical abnormalities, and they have lower triglycerides. So again, you can sort of see where some things we learn in flies are also going to be true in mice, and these are the kind of insights we often use. For those of you who are in our study of some of these proteins at the NIH, you know that we check triglycerides, we check cholesterol, we check things like that. And we're doing it because we have indications from our other studies that it might be something that's relevant for the human disease. So again, I think we learn a lot from these studies, and we directly apply a lot of this knowledge as we're studying, you know, how this affects people. So again, if we go back to the ER proteins, we see all these different ER proteins affecting this thing. What if we knocked out more than one of them at the same time, or if we mutated more than one? And this gets back to the point I think Peter raised earlier, is in some cases, let's say you make a mutation and you don't get a phenotype. Well, there's a couple of explanations. One is you don't have the right mechanism, as he was arguing for SPG4. However, another possibility is that it's the axons in a mouse aren't long enough. You know, a person, if we're talking about a 1 meter axon in a human, the mouse isn't even that big, and their corticospinal tract isn't the same as ours. So they don't have, there's some things that are different about it. So there's the chance that we might have to actually make a more severe form of HSP to see something significant in the mouse. So what we've done is we've made 6 or 7 different mutant forms. I won't show them all, but the one that has the most prominent abnormality is the one where we've made a mutation in it last. And then this is like what Peter mentioned with these gain of function. We didn't take the gene out. We just changed a residue that made it look like, that made the protein not work. And in fact, almost all people with this mutation have the same kind of change. They have a amino acid substitution that impairs the protein function. However, in the case of REAP, in most patients with that do have what we think is haploinsufficiency based on several of the mutations. So instead of just haploinsufficiency, we've taken it away completely. And there are a couple individuals that do have, you know, the protein completely lacking and have even a more severe form of HSP. So again, that's probably correct. What if you combine those two? Well, they both bind, the proteins interact with each other. We think they work in the same pathway. So our hope is that we've actually created just a more robust or more severe model of HSP that can be tested more quickly, maybe. And understanding the limitation that no person actually has this genetic change, we're hoping that we've recapitulated the main features and they would respond the same way. And if you look at these guys on the top, you'll notice that the mice move just fine. However, look how much more severe this one is. You'll notice the hind limbs, and you'll notice the obvious spasticity, you know, the shaking and really trying to pull. It really looks like a paraplegic mouse. So I think in some ways we have recapitulated key features of this. Oops. So this really leads directly into models to develop and test new therapeutics. And again, this was brought up by both speakers before me, I think very eloquently, and how important this is. You know, we can't just go from a cell to a person usually. We really do have to understand not only how mutations or how interventions we do might be affecting different cellular processes, but how also that they improve meaningful phenotypes. You know, we don't want to just know that, well, we've corrected the signaling pathway or corrected the ER. Does that actually make a person better, or does that make an animal move better? You know, those are very important things. And one advantage of this system that we've developed is, as you'll notice, the age. We see in rotor rod, ledge test, hind limb clasping, these are different tests that you can do to assess the function of a mouse. And if you'll notice on the far left, it's a latency to fall. How long does it take you to fall off of a spinning rod? And you'll notice each one of these is an individual mouse as it gets older. And you'll notice all the orange ones stay on pretty long. All the mutant ones are the ones I showed you with the double mutation. almost every single one of them after 8 weeks. In fact, every single one is worse than every single wild type. So it's a big difference. And this kind of big difference is the kind of thing that we can use to do therapies. And in fact, we're starting to do that. You'll notice the same thing with ledge test, hind limb clasping. And what's important about this age range is from 4 to 24 weeks. It's basically from about a month old to 6 months old. So the mouse is showing a lot of abnormalities at an early stage. And since at age 4 weeks they're adults, we can really start treating them pretty easily at this age. So currently we're trying several different therapies to see, what if we can make these mutant animals much more like wild type. And the advantage, I think, of this system is that it's fast. We can see these, the change from 1 month to 6 months is dramatic for a lot of different types of tests. So we're hoping our intervention will see that we have enough of an abnormality in the mutant forms that we'll see an improvement. Rather than having a weaker mouse model, you might have to wait 1.5 years or so, and then they're getting old. And then again, then you're not really sure, you're running into aging, interfering with a lot of the phenotype. So I think this could be a very useful system. Again, with the caveat that we have created a mouse that doesn't replicate any person's genotype. In other words, they have more than one HSP at the same time. Though, as Peter mentioned, that might be more prevalent than we think. Often when we make a diagnosis, we don't look at the other genes. It may be more common, in fact, that some people do have, if not as overt as the case of this mouse, that they could have multiple genes contributing together. And that could be a reason that it's harder to diagnose in some cases. But I do think this is a valuable animal model for the testing of compounds that we'll have. Another advantage that we have with this model is that we can see a specific abnormality in the corticospinal tract. So the tract has these nice myelinated axons of different sizes and shapes. And look at how disordered it is in these mutant animals. They don't look right, and they're smaller and shrunken. And if we zoom right in, we see that there's ER normally in this axon of the corticospinal in the wild type. However, look what happened in the mutant forms. The ER is going the wrong way. It's supposed to be going longitudinally, and now it's going transverse. But what this is really, so we now have another thing we can look for. So we start treating them. Well, what happens if we look at 6 months? Do we see this, or did we make it go away? So it's important to look at both. Have we hit our target? Have we intervened in the cell itself where we think we're intervening? And second part is, have we made the animal better? Have we improved the symptoms? And both are important. You want to know if you've engaged your cellular target, but you also have to see improvement as well. So one of the things that we've been working at over the last several years, and this is sort of the show and tell part of my talk, is that we felt that we were limited in seeing the abnormalities because we couldn't see them in enough detail at high enough resolution. So if you think about looking at these cell structures, they're very small. And microscopes are good, but in some cases they're not good enough. We're trying to see structures, but if we can't see it at the level that we need to see an abnormality, then we'll miss something. We'll miss a function. We'll miss how it's really working in a cell. And this probably happens, to all of us in the morning if you wake up and look at the alarm clock and it's blurry and you try to guess what time it is. But you'll sometimes be right and you'll sometimes be wrong. But what if we could have your glasses on beforehand? You'd see it and you'd be right all the time. And that's what we're trying to do. We're trying to make the cell less hazy and see things at a higher resolution. And so we've been working with a group at the Howard Hughes Medical Institute at Genelia Farm. And these are probably 3 of the best cell biologists on Earth. In fact, Eric won a Nobel Prize for what I'm going to show you. So we've really been able to really have a unique opportunity to really work with the best cellular imaging that's probably anywhere in the world. With Eric, Jennifer, and Harold. And I'm going to show you some different things that we've been doing with them. First is we've been using new kinds of imaging technology. And one of the problems with our eyes and with any kind of optical imaging in many cases is it relies on it has the limitation of diffraction. And there's many methodologies that are out there now that overcome that limitation in different ways. And I won't go into all the physics of how that's done. But what I think is important is it allows us to see the structures at a much higher resolution than was ever possible. And these are some of the methods we use. They all have acronyms. You don't have to know any of them. But what it essentially does, it allows us to see things at higher resolution than we ever could before. Here's an example on the right. If you look at it, what we would see with really one of the best scopes that you can buy at the top is blurry. But if once you actually get higher resolution, you can start to see holes and other structures that you couldn't see before. And it becomes important as we try to understand how proteins function and how they affect organelles and other compartments with the cell. If we can't be sure of what we're seeing, it's very hard to know if we're right or how something's affected. So I think it does emphasize that it is important that even something as fundamental as what we used to call an ER sheet actually isn't a sheet at all when we can see it at higher resolution. And here's another example, and this is using a technique that Harold Hess pioneered called focused ion beam scanning electron microscopy. And many of you may have heard about EM. Some of you probably had biopsies or something and they've shown EM. One of the advantages of EM is it's very high resolution. One of the disadvantages is you have to cut sections, and you can only slice a loaf of bread so thin. But if you adapt a new technology called focused ion beam, you don't have to cut. You can take a block of tissue that's been fixed in some way or frozen and actually polish off the surface, image, polish, and so on. And that gives you a far, far greater resolution than you could ever get with conventional microscopy. And this is an example of what you can see. So if you do this, you essentially see this structure appearing, and when you recombine the entire thing, you get an image of what the ER looks like. So again, we're picking the ER as an organelle to talk about, but remember, this could apply to a lot of other organelles as well. And look what happens when we reconstruct it. We see this three-dimensional structure that's very complex. However, if you would have looked at this with a regular confocal microscope, you'd have seen what's in white. It looks completely different. So again, I think it emphasizes that traditional microscopy, it's important to remember that we're not always seeing things as they are, and it's important to try to get the highest resolution possible where practical. Of course, this comes at a cost in terms of time, money, and computing power. So sometimes it's not practical, but it's important to remember that we do have this ability, at least at some level, to analyze some of our model systems using these very, very high resolution techniques. To the point where we can see structures like in C, these like honeycomb-like structures of ER that before we published this in 2016 really weren't described. You know, we're still learning about the cell. As we can see things at higher and higher resolution, we're actually seeing things that we didn't know existed. So let's go back to our mutant form. How can we use this technology to give us more insight? We know that this ER looks abnormal, but ER is continuous. And of course, in a EM section, we can't see the continuity. We just have one section. And we can't cut sections because the ER is really thin and the cut sections are too thick. You can't reconstruct it. But what if you could do fib-sem, that really high resolution imaging on an intact spinal cord? And this is what you see. The wild type, this is actually a spinal cord, a corticospinal tract of the mouse, and we've reconstructed the ER. And just to show you some of the myelin and other structures, and look what happens to the mutant. Those are those transverse structures. And in fact, these exist other parts of the cell at certain times. So it's not a completely abnormal structure. It's just in the wrong place, and there's too many of them. And we can even reconstruct mitochondria and other organelles. As Peter mentioned, there's these, sometimes you see these sort of swellings in axons, and it'd be nice to know what's in them, and to also, as you go along the axon, see how the organelles, see what's disrupted. And we have the ability to do that now. It's just we have the ability in a very, very limited capacity, because to do this is a huge amount of work, and it took one student in our lab months to do just this one axon. So how do we get around that? I mean, we want to know this information, but yet it's so hard to do by hand. And we've gone to what's called artificial intelligence or machine learning. And you can see that right now people still do a better job than machines. you can see on the left that we do see the ER, we see the mitochondria, and this is a much bigger axon, so this would have taken us like a year to do. And the machine, of course, can do it in a couple minutes if it's trained properly. But you notice that as we do our mutant forms, you start to see some abnormalities, but it's not quite as clean. So we're still working with a company and doing a lot. We're trying to come up with ways to do this automated. And when we can do this automated, we'll be able to get a huge amount of information out of it. But we're still teaching the computers how to read the EM images. And it's not easy because sometimes the contrast, telling one organelle from another is difficult, and we can only do it based on morphology, by appearance. There's no way you can stain or anything like that. So this is the kind of thing I think where, again, we always say that how much advance there has been in terms of genetic analysis using these, with these advanced computational methods. We're going to be able to use the same thing for cell biology. It's just that there's still some bugs to work out. We'll eventually get this to work. We're still, I think in a couple of weeks, we might be able to get the one on the right to look better. But I think we want to emphasize that look at all the organelles we can see and the resolution we can see. These model systems, as we're able to really investigate them, will give us a tremendous amount of not just phenotypic or like behavioral information, but also cellular information in the exact axons that are affected in this disease. Another thing to think about is things are in motion. Many of us study sections, you read papers and you'll see a picture of something. Remember, it's just like taking a picture of a person. People are moving and so are cells, and so is every organelle within your cell. And that movement can be important for disease. And many times we miss it because we don't look. So this is what the ER looks like in real life. Everything I've showed you is moving or was still, but this is what it's actually doing. It's moving like crazy. And that movement ends up being important for disease. Here's an individual protein. We now have the ability to track a single protein molecule as it's moving. And this is an ER protein. It's sort of peripherally involved with one of the forms of HSP that we're studying. But you'll notice that each one of those movement or the sort of events where the protein goes to the ER. So the ER is in blue and this protein is in the yellow at the top. We're able to see different types of movement. And it turns out that that movement is very important for understanding disease in some cases. And here's an example I know that Peter would like because it's spastin. And we know that if you make a mutation in spastin, as he, I think, really nicely showed, you get a different type of effect than taking it away. So what if you make a mutation in spastin that causes HSP? What happens to how it moves? And this is the M1 form. So it's actually in the endoplasmic reticulum. It's in the membrane. And this is what the mutation does to it. It's not moving anywhere. Look what a normal one does. So it moves all over the place. So this is just one spastin molecule. But when we do these studies, we can actually do hundreds of thousands at once. So if you actually look at that many, we can see that there's this dramatic effect in mobility. So again, something we hadn't really thought about, but important to consider as we consider how disease mutations affect proteins. Again, if it's not there, we can't look at it. But in this case, you can see a normal spastin protein has done all that movement. It's gone all the way around the ER, and this other one didn't budge. Probably because it's locked onto a microtubule and can't move. But so we've really changed the mobility of organelles, and the potentially the way that spastin functions is by severing. Well, if it can't move, it's going to have a lot more difficulty interacting with other proteins or severing the microtubules if it's stuck there. So again, these are, I think, another example of how dramatic these changes can be. Oops, that's just going again. And here's another example. This isn't HSP, it's ALS. But it's a mutation that causes abnormalities in ER structure, and specifically how the ER functions with the mitochondria. And I think Peter made a very nice point. Is, we really, if you're thinking basic science, you don't always think, well, I'm only going to study this disease. You think, what other proteins are involved in the process? And many times you'll have the same process be involved in different types of neurological diseases. So it's important for us, as we do this research, to think not only like a clinician, but also like a basic scientist. And here's an example of a protein that, again, causes a form of ALS. And it actually moves along the contact site between the ER and the mitochondrion. However, if you look at a mutant form, again, just one point mutation, it doesn't move anywhere nearly as much. It's stuck. And so sometimes we can learn a lot about, and this is just, again, an individual protein. So our ability to image and to understand the basic biology of our diseases and the changes that occur in these genes has really, really, sort of exploded, much like genetic testing has over the last few years. The only limitation really for this being more widely used is it's very expensive, very hard to do. It took us years to get the tracking to work. But now that it's working, I think we can apply it to a lot more different diseases. But there's a lot of technical challenges with these, but I think it is ultimately going to be very important. And again, we're applying it to the HSPs as the first diseases we're studying. So hopefully we'll get insights that build on all the other insights you've heard about, and that will allow us to really, as I think Peter really made that point very strongly, and it was correct, is we really want to understand what our target is going to be and understand that that target is a meaningful one for treating this disease. Because, we certainly don't want to keep pushing things through that fail, because then our pharmaceutical partners, drug development, they lose interest. It's important to have as strong of a case as possible as we move forward. And I think by combining model systems, by combining, understanding a lot of these new cellular tools, we have so many genes that creates a tremendous advantage for us. And so again, the last step is going to be, we have model systems to develop and test new therapies. We understand the biology. We have all this genetic information. We have different types of model systems where we can really sort of compare and contrast them. But ultimately what we want to do is, and we've tested the models, we maybe have some good ideas for therapies, is we want to conduct clinical research studies. We want to make sure it's safe and efficacious for new treatments in people. And I thought Darius did a great job of showing some of the things that are necessary. We have to understand the diseases, which is why he looked all over the world and really tried to compare and contrast how people present with this in different areas and what might be the type of symptoms that we want to look at to go along with the sort of cellular changes that maybe we understand more. It's also very important to understand different phenotypes that might be important in different types of HSPs. But of course, we also have this unifying abnormality around the spasticity in the lower extremities, the gait disorders, and the so on. So I think it's going to be, in many ways, and again, you've seen lots of nice examples of it today, we have a lot of insight into mechanisms, and we're getting more and more of them. We're hearing ideas about different treatments. And now we need sort of that infrastructure for clinical research studies. So as Frank mentioned at the beginning, one of the things that I'm really interested in doing is sort of developing that more when I move to my new position. So again, I think Peter already made this exact point, but I wanted to emphasize, because I think it's really important. When we think as clinicians, we're often thinking parts of the body, we're thinking circuits, we're thinking how patients present with different symptoms. However, it's important to also look deeper in terms of how these proteins are working and understand really the basic biology of what it is, and not just limit ourselves to the symptoms that we see in terms of therapy. And again, this is sort of another example. You can have many different diseases that are all caused by abnormalities in ER shaping. So it's important for us to understand the clinical manifestations, while at the same time, it's important for us to understand that basic biology and how those two relate to each other, so that in the future, when we think about these targets, we might not think of just HSP, we might have other disorders that we can bring into the fold. And that can be very important for generating more interest, because the population that could potentially benefit would be greater. And we might also be able to incorporate some other types of disorders into different types of trials. So again, I think that both the previous speakers really made the point that I was hoping to make, and they'd already made it, is really that how important it is to do the clinical studies, how important it is to do those basic studies, and really how to bring them together. So again, my new position, I'm going to be the head of a very large division that works on different diseases, from Parkinson's to Huntington's, different types of tremors, dystonia, and of course we're going to add HSP to that. So certainly as time goes on, for those of you who know me or want to drop me a note, if there's anything you think about that would be a good thing to start to incorporate into this, let me know. Ideas that you'd have, like if you see your doctor, what do you wish you had? Are there types of relationships with physical therapists or with other divisions of medicine that we could do? We really have pretty much a blank slate as we set this up. And again, there's spectacular people at this hospital. So we have a really unique opportunity, I think, to build a new center for HSP. And again, any ideas that you have, pass them along to me. Again, it's harder to do this in the setting of a coronavirus epidemic because we're not able to see or do anything like we normally did. But certainly over time, we're going to have the opportunity to create something. And certainly part of my vision is to make sure that it's not just for patient care. And again, that's in contrast to the NIH. Really, our goal here has been only research. We really want to make patient care an important part of this with the therapies we have now, but we also want to take advantage of the Mass General Hospital's real strength in a lot of clinical research. And they've been doing many, many trials with neurological diseases over the years. And we're really hoping to tap into that expertise and infrastructure, that as all these great ideas come up to the point where we can, test them in people and to see if we've got something beneficial, that hopefully we'll have a better infrastructure than we have right now to do that. And that's certainly my hope for going here and really helping develop that next phase of HSP research, because I really think that after 20 years in this field, that there's been so much progress in the basic sciences that we now see all that starting to filter into new ideas for therapy. And I think now is the time where we think of, again, as Darius has already been doing very nicely for his disorder, maybe thinking even more broadly in terms of how we can create the types of infrastructure that we're going to need for testing, therapies in a rare disorder. And again, Darius brought up some of the challenges that we have, but there's also a lot of strengths too. And I think that it's important to start thinking about that now, because I think we're sort of entering that phase where therapy is going to be the next thing we're thinking about, how we translate all these great insights that people are making into ways that we can actually treat people. And again, I think we're sort of at that cusp now, and the next phase of HSP research, I think, is going to increasingly be those early stage trials where we really want to use the best ideas we have and test them the best way we can with the best type of trials. Again, we're not going to get the same number of trials, the same amount of money, the things that Alzheimer's, Parkinson's gets. And that's why I think it's even more important that we're really smart about how we design and pursue these trials in the future. And again, this is like I think everyone else has shown, these are very collaborative studies. We work with a large number of people in basic science and clinical medicine, throughout the US and the world. That's a picture of my current building. And again, I just showed you sort of at least a picture of one part of our new center, which we'll be moving to in 2 months. And I just wanted to, again, thank you all for listening. And as Peter, I think, did, they've said that any questions that you have, you're welcome to write down and put in the chat. And we're going to arrange a session later where maybe we answer some of the ones that have come up the most frequently, or some of the ones that might be, important concepts maybe that we didn't cover, in quite as much detail, or that was a little bit more confusing. But again, I certainly want to thank SPF for allowing me to speak again this year, and to all of you for listening. And again, I hope that we're always here, and that if there's any questions, particularly through the chat, that you can get to us, we're certainly happy to take a look and answer them in a future web meeting. I think we might do more of these now that it seems like it's been so successful. Again, thank you again.